All right, I'm calling this Enzymes Part 3, talking about activators and inhibitors that help us control as on-off switches. The enzymes in class today, I had you fill out or draw this overall outline about activators that turn enzymes on or make them work, and inhibitors that block enzymes from working. And so I'm going to go back up in the notes and talk about each of these. And you'll notice I also wrote here non-competitive inhibitors are what we call allosteric inhibitors. We're going to see they bind to something called an allosteric site and change the shape of the enzyme. And activators can also be allosteric. So I'm going to start just scrolling back here to this part in the notes and just go into all of this in a little more detail, but that's the outline. Okay, so again here we're talking about activators and inhibitors, things that the on-off switches that can either make enzymes active or block them. Some enzymes have something called an allosteric site, okay, and things called allosteric regulators can bind there and change the shape of the enzyme. So allosteric regulators can be activators, or this could be an activator or an inhibitor, which I'm going to show you down here in this picture. Okay. First, let's look at this picture. If this is my enzyme, and this is my active site where the substrate binds, or the substrates bind, an allosteric site, by definition, is a site other than the active site. So there might be another binding site somewhere on the enzyme that either an activator or an inhibitor can bind here. And once it binds here, it's going to change the shape of the enzyme, okay, which is going to affect the active site either in a good or a bad way. So let's look over here. So this is allosteric inhibition. So let's say this is my enzyme. Here's my active site. And I wrote it's working. So this is the right shape. The active site is the right shape. But if I want to turn this off and inhibit it by allosteric inhibition, which is also called non-competitive inhibition, let's suppose this little green molecule binds to the allosteric site here, and you see how the enzyme changes its shape, and the active site now isn't the right shape. So the substrate won't go in there, okay? So it's working. A non-competitive inhibitor changes its shape so that it's not the right shape, so that it's not working. So it's still blocking the substrate from coming into the active site, but it's called a non-competitive inhibitor or an allosteric inhibitor because it's not competing, it's not binding in the active site, it's binding here on an allosteric site. Down here, this would be the same thing, but this is an activator that's an allosteric modulator. So in that case, I might have an enzyme that's not the right shape. And sometimes we do this on purpose. Like when it gets made at your ribosomes, the enzyme tertiary structure or quaternary structure just isn't the correct shape to start out with. So my active site isn't right. And I might want it that way so that I can turn this enzyme on when I need it by having an allosteric activator. And again, this could be something we call a coenzyme or a cofactor. We talked about this today. If it's an organic molecule, like a vitamin, that's doing this, we call it a coenzyme. If it's an inorganic ion that we call minerals in our diet, we call it a cofactor. But both coenzymes and cofactors, vitamins and minerals, act like these activators, and it could bind here in the allosteric site, but in this case, it's changing the active site, and now the enzyme's working. You can see the substrate is in there. So a non-competitive inhibitor and some allosteric activators are binding to the backside of the enzyme and changing its shape, either so it doesn't work, or if it's an activator, it's changing it for good so that it does work. Okay. So whenever I talk about an allosteric you hear that word allosteric, it means the protein is changing shape. Something's causing it to change shape, okay? Okay, so this is talking about, again, activators again. Some enzymes require activators. I talked about this in class. They're either cofactors or coenzymes, which we can see down here. A cofactor is something that's inorganic, like a metal ion, like some of these things we need, like calcium ions, magnesium. These are ions that we need in our diet, minerals. But because a lot of them are going to help an enzyme work, and a coenzyme would be things, vitamins, like, and then these other ones, which we'll learn about later, but the vitamins you know of are things like, vitamin A, vitamin C, vitamin B12, things like that. All of the reason you need those in your diet is because they are either cofactors or coenzymes to help certain enzymes function. A couple little just terminology things, they make it difficult here. I'll do this in green. Some enzymes, like simple enzymes, just work all by themselves, okay? Enzymes that require an activator, we call them conjugated enzymes. And that means they're composed of, in order for the enzyme to work, you have to have the enzyme by itself, which we call the apoenzyme. And then one of these two things has to bind, a cofactor or a coenzyme. So let's say this is in a picture, this blue thing. This could be I'm calling this an apoenzyme. I think that got cut off on your notes. That's the enzyme by itself. And maybe it also needs to have a cofactor and a coenzyme, like a mineral and a vitamin, that has to bind here to create the correct active site. So I would call this a conjugated enzyme because it needs to have a cofactor and or a coenzyme, or both, bind in order to have it function. So you'll notice here two things. Activators, this picture is showing them binding in the active site. So they're kind of creating the correct active site. So some cofactors and coenzymes, or some activators, they're not allosteric. They're binding in the active site, but they're helping to shape the active site correctly so it can work. Other activators might be allosteric activators, which mean they're going to bind an allosteric site, which is some other site, like let's say they might bind back here, which changes the shape of the enzyme so the active site works. So some activators are allosteric and some might go right in the active site. Both are helping the enzyme be the right shape. Okay. So just a little bit about, I mentioned that activators, they're either vitamins or minerals, and we're not going to get all into this. If you ever take a nutrition class, I don't like the color, there we go. Like when we talk about vitamins that you need in your diet, or minerals, calcium, iron, iodine, and if you don't have a certain vitamin or mineral in your diet, you often get like you have a deficiency or a disorder or something's wrong with you. This tells you the symptoms. And I'm telling you, this is an example of like vitamin A is a coenzyme for an enzyme in the eye that needs vitamin A to work. I don't want you to know that, you don't have to know the name of that. But so you can see that if you have a vitamin A deficiency, because it's helping an enzyme in the eye, obviously something in your eye, some enzyme is not working. So you can see that the symptoms you end up with are sort of problems with vision because there's an enzyme in the eye that's not working. So you could look up for each vitamin, like what enzymes do they help activate? Like vitamin B1 is a coenzyme for an enzyme that's in cell respiration in the mitochondria. And the mitochondria, right? Cell respiration in the mitochondria makes energy. So this is what makes energy for cells in the form of ATP. We've talked about that. So vitamin B1 is a coenzyme for, I think there's several enzymes in the cell respiration process that if you don't have vitamin B1 there, you're not going to do cell respiration in the mitochondria. You're not going to make ATP, which is energy. So you'll notice weak muscles, little energy tends to be the symptom of a vitamin B1 deficiency, and they call that beriberi. Vitamin C deficiency is called scurvy. I do not want you to memorize. I'm not going to ask you on a test to memorize like which vitamin causes which deficiency or which symptoms. This is not something I want you to memorize. I'm just showing you that kind of examples in your real life of why we need vitamins and minerals. They make certain enzymes in our body work. And if they don't work, if that enzyme is deficient, you have symptoms that relate to that enzyme. Okay, that's all I want you to know about that. Don't memorize any of that. Going then back to inhibitors, I told you they can be competitive or non-competitive. So they're both defined here. And there's a picture here. So this is a competitive inhibitor. Here's my enzyme in purple. A competitive inhibitor just binds right in the active site and blocks the substrate. I say it takes the parking spot of the substrate. A non-competitive inhibitor, which could also be called an allosteric inhibitor because it's going to be changing the shape of the enzyme, binds in a back site, like an allosteric site, and that changes the shape of the enzyme, shifts some of the weak bonds, and its shape changes. And then the active site isn't the right shape, so the substrate can't go in there. So they're both blocking the substrate from getting in there. This one directly by going in the active site, this one by just binding an allosteric site and changing its shape. Okay. So how would we be able to tell? So this comes up a lot on the test, on tests, wink, wink. How would I tell if I know that a certain enzyme has an inhibitor that's blocking it? How would I figure out if it's a competitive inhibitor or a non-competitive inhibitor? And I wrote that in a competitive inhibitor here, if I add more substrate to the cell or the test tube or whatever I'm testing, it can outcompete the inhibitor. Whereas if you add more substrate, it can outcompete the inhibitor, but a non-competitive inhibitor, you cannot outcompete it. So for example, if I had, so I think of this graph down here, it might have got cut off on your notes. Okay, so let's say here's my substrate concentration and I'm measuring reaction rate. And my blue line is just my normal. I put enzyme and substrate in the tube. Remember, it goes up to the Vmax and it's going to level out because there's a certain amount of enzyme in there. If I add a competitive inhibitor, that's what that says, you'll notice that the reaction rate in general is slower because an inhibitor slows down the reaction. But you'll also notice that as there's more substrate, it starts to outcompete the inhibitor, the competitive inhibitor. Whereas a non-competitive inhibitor, if I put enzyme substrate and a non-competitive inhibitor in the tube, it changes the shape of the enzyme so that even if I add more substrate, it's still not going to work. It doesn't matter. So this one I can outcompete. So if I just keep adding more and more and more and more of this, it's like a competition for the parking place. Eventually, this can like outcompete, and you'll have maybe a few enzymes that get blocked, but more enzymes that substrate is binding to. So that's how you can tell. If I gave you a situation and I said the enzyme's not working, there's an inhibitor in there, figure out if it's a competitive or a non-competitive. What you would do is add a ton more substrate to that test tube and see, measure product or something, see if the reaction rate goes back up. If it does, it's a competitive inhibitor, and this is just outcompeting that. If it's changed, if the inhibitor is non-competitive and it's changed the shape of all the enzymes in the tube, I can add as much substrate as I want. I'm still not getting any reaction, and so graphing it would just look like a straight line. Okay, so that's enzyme with substrate and a competitive inhibitor, enzyme with a substrate and a non-competitive inhibitor. So that graph shows up a lot to be able to kind of analyze that. So this just is, this is just a silly one. Like if the blue was the control group, if I was increasing substrate concentration, like if this is my control, activator, you can see increases the reaction rate, inhibitor slows it down. This would obviously be a competitive inhibitor because a non-competitive inhibitor would be a flat line. So that's kind of what it would look like graphically if I had a control and then I added an activator to that enzyme. You would just see the reaction rate speed up. So just get used to looking at graphs. We're going to practice some in class next time. So we'll do some practice AP questions where I give you graphs and you kind of analyze that. And I think so we went over that outline. I'm going to come back to negative feedback here in a second. I drew this picture. It's the same as I don't know if this helps you. You can stop and copy it or take a picture of it. But it's showing the difference between a competitive inhibitor in a picture, a non-competitive inhibitor, which is an allosteric inhibitor, or an allosteric activator. Kind of the same thing. I think these pictures are simpler. Here's my enzyme, active substrate fits in there, so does competitive inhibitor, right in the active site. If I have a non-competitive inhibitor, here's my enzyme, here's my active site where my substrate would fit. Once it binds to the allosteric site, you can see now the active site has changed when the inhibitor is bound. So the substrate doesn't fit right there. And then an allosteric activator is the opposite. My active site, notice, doesn't fit the substrate, so this isn't working until the activator binds the allosteric site, changes its shape, and now the active site does fit the substrate. It's just kind of a more simpler picture I do sometimes in class, but we didn't have time. A couple other examples, just real-life examples of inhibitors I wanted to go through. And I think you have this written in your notes. Gray hair. Why do we get gray hair as we get older? Well, in your hair, you have a brown pigment called melanin, and during your lifetime, when you're younger, there's sort of an enzyme pathway. There's actually multiple enzymes in a metabolic pathway that produce and make this brown pigment in the hair follicle, okay? So when you're not old, all these enzymes are working, and you're making melanin, and your hair is brown, let's say, okay? As you get older, hydrogen peroxide is actually an inhibitor of this first enzyme. So the first enzyme in this pathway is something called tyrosinase. Don't memorize the name of that, but it's an enzyme that you need all the other enzymes. But what happens as we get older, remember we talked about the enzyme catalase? Catalase breaks down hydrogen peroxide. So catalase, as you get older, you have less of it. So if I don't have catalase to break down hydrogen peroxide, which all cells, this is a waste product that all cells make, and it's toxic. But as you get older, the catalase, you have less catalase to break down the hydrogen peroxide. So the hydrogen peroxide acts as an inhibitor to tyrosinase, which is in this pathway to make pigment. So you're basically not making melanin anymore when you get older, and gray hair is just basically without pigment, what hair looks like with no pigment. So I just thought that would be interesting. A couple of other times where this would apply. Some drugs that we use are inhibitors. Okay, so inhibitors are bad, but we use them. A lot of antibiotics, here, these first two examples are antibiotics. So a lot of the antibiotics that we've discovered and use are inhibitors that block a certain enzyme in bacteria. So we want to make sure we're blocking an enzyme that bacteria use so we can stop them from multiplying, but not one that we use, because that would backfire on us. There's a bunch of antibiotics. If you look it up, there's a whole list of a whole bunch of names that are called sulfa-drug antibiotics. And the way these work is there's an enzyme in bacteria that makes folic acid. Folic acid is something they need. And so a lot of these sulfa-drug antibiotics are inhibitors. When you take them, they're blocking this enzyme in bacteria cells because we don't have this same enzyme. And if the bacteria can't make this needed folic acid, they basically start dying off and they can't reproduce. So that's how we kill bacteria, by using an inhibitor that blocks them. Another example, penicillin, which was one of the first antibiotics discovered. What it is a chemical. It's just an inhibitor that blocks an enzyme in bacteria so that they can't make cell walls. I don't know if you remember, bacteria cells have a cell wall too, kind of like plant cells. And so if they can't build cell walls as they divide, they just they can't divide and multiply. And essentially that slows them down. So those are just examples of antibiotics that are inhibitors in bacteria. Here's a couple other examples of inhibitors, right? So these ones are helpful to us because they block enzymes in bacteria. These are some other inhibitors that I just wanted to mention here. We talk about mercury and lead as being really toxic and bad to us. The reason, like heavy metals, mercury and lead are heavy metals. These are inhibitors there's lots of enzymes in our body that if you're exposed to a lot of mercury and lead, they're blocking a lot of enzymes. So there'll be a lot wrong with you. That's why those are bad. Nerve gas, which you might have remembered, nerve gas, they used in, I forget, like in the war. And basically people, when they'd spray this nerve gas, would just suddenly be paralyzed and freeze up and then die immediately. And it's because it's an inhibitor that you breathe in, but it's an inhibitor that inhibits this enzyme acetylcholinesterase, which is an enzyme that is part of nerve and muscle functioning. So essentially, we would kill people by spraying nerve gas, or I don't think we did it. I forget who used it the most, but they would spray this nerve gas, and you'd breathe it in. It would block your nerve and muscles from functioning by being an inhibitor to that enzyme, and that's what killed people. Cyanide is also, we know, is a poison that kills people. It's a toxin, and it's because it's an inhibitor of this enzyme, cytochrome oxidase, which is one of the enzymes in the mitochondria, again, doing cell respiration. And remember, this is what produces ATP energy that we all live off of. And as soon as, if your mitochondria aren't making ATP energy, you're dead. You can't fight entropy. So I don't want you to memorize. I'm not going to ask you to memorize like acetylcholinesterase or cytochrome oxidase or mercury or but, I might have like a question on a quiz that says something, like true or false. Some antibiotics are inhibitors to enzymes in bacteria, and you would say true. Again, I don't expect you to memorize the details of any of these examples. I just want you to know in general kind of how this applies to kind of our world. Make it interesting, okay? Again, I'm not going to ask you to don't memorize tyrosinase, but, gray hair is basically because something's inhibiting an enzyme in our hair that makes melanin. So that's it. I guess negative feedback. This is really simple. I might as well just do it right now. Feedback inhibition, also called negative feedback loops. This works a lot in living things. Okay. So many metabolic pathways. This is how they're controlled. And I mentioned today that we often have not just one enzyme reaction, but we'll have like multiple steps to a metabolic pathway. So like threonine gets turned into that molecule by enzyme 1, and then enzyme 2 will turn that molecule into that molecule, and enzyme 3 will turn that molecule into that molecule, and so on. And let's say this is a metabolic pathway, and the whole point is to make isoleucine, which you may remember, threonine's an amino acid and isoleucine's an amino acid. It's just an example of a metabolic pathway. But the way this would be controlled is often the very last thing at the end of a metabolic pathway. It's isoleucine does something. We're going to use it as an amino acid to build proteins. But it's also, okay, so that's why I guess there's two lines there. We're going to use it for something, for building proteins, etc. But some of it will also be an inhibitor to the very first enzyme in the pathway. And that makes sense, right? If I already have a ton of isoleucine in my cell, I don't want to waste energy, time, resources running this metabolic pathway. Okay? So it's kind of like your thermostat. Like you set it for a certain temperature, okay, like your heater. And once your heat gets to 70 degrees or whatever you set it for, the heat shuts the thermostat off. So that's what we call a negative feedback loop. And that's often how a lot of pathways are controlled. The final product does something on its own, but it's also going to act as an inhibitor, and it's usually the first enzyme. Because if I blocked enzyme 3, then I'm still wasting energy doing these first two steps. Once isoleucine then goes low in the cell again, then there's going to be less of it. It's going to come off of that enzyme, and that enzyme will become active again and so forth. So that's a negative feedback loop, also called feedback inhibition. And that is the rest of the enzyme notes. That is it.